Okay, great.
Okay.
Okay, well, thank you very much for inviting me to speak with you today. My name is Jennifer Baltzegar, and I'm currently a postdoc at NC State in the Genetic Engineering and Society Center. But I also got my PhD here in genetics with a minor in genetic engineering and society. And my main areas of interest are population genetics and population structure, evolution of insecticide resistance, and then the use of genetic engineering and its implications. My primary work is actually on mosquitoes, specifically the species that transmits dengue, Zika, yellow fever, chikungunya. And so I actually put the reference for that, my most recent paper that's published there. But today I'm going to talk about, if I can change my slide, there. Work I did during my PhD on maize weevil, which is currently in review. And I think this presentation will get linked to that preprint at some point. But so the topic is developing genetic resources for and determining the population structure of maize weevil in southern Mexico. And so I'll start with some background. I know this is a maize evolution group, and so my goal is to get people to really appreciate the maize weevil and its importance in food security. And so this is the close-up of the maize weevil. The scientific name is Setophilus zea maize. And as you might gather from its name, both the common and the Latin, it is a pest of maize. And it is a worldwide problem, although in the US and probably other developed countries, it's not a big deal because we have money, we have ways very easily that we can control it. But in subsistence communities, especially, it can really cause negative economic impacts and actually be a really serious threat to food security. There have been reports that up to 100% of grain has been lost due to the maize weevil. And although they are called maize weevil, they can target other cereal crops. Now, there are a couple of related species that look very, very similar. You need a microscope to tell them apart. And although they look so similar and they share similar habitats and niches, their zea maize's closest relative, the rice weevil, which is Setophilus oryzae, is about 2 million years diverged. So pretty strong reproductive barriers exist, and although they still look the same to us, they are quite distinguished from one another. There is sexual dimorphism, and you can tell the females have a longer, thinner proboscis, and the males have a shorter, stouter proboscis. And it's this proboscis that allows them to really wreak havoc on the maize. So the female, when it's time to lay eggs, will use her proboscis to eat a hole into a kernel. She'll turn around and she'll lay one egg. She'll turn back around and seal it up and make a window into the kernel. And that egg will then hatch and develop within the kernel until the weevil is an adult. And all this time as a larvae, it's eating the pulp inside and basically destroying all the nutritional value of the kernel. And this video that's looping on the right-hand side is actually a newly halfway emerged adult coming out of a kernel of corn. And so the maize weevil originated in Southeast Asia about 8 million years ago. And if you're thinking, why is it the maize weevil? It's 8 million years old. It's from Asia. Maize evolved in Mesoamerica about 10,000 years ago, at least as we know it. Well, historically, maize weevil was actually a pest on like acorns and other types of tree nuts in the forest. But once maize was introduced to the rest of the world, it was just the perfect host. the corn was just like great for the maize weevil to use, and it then developed this really close relationship with maize. And as I said before, in developed countries, maize weevil is easier to control, but in subsistence farming areas where people are storing maize in a variety of ways, it's a problem. And pretty much almost every farm I went to had maize weevil. It was the most common pest I saw when I was working in Mexico. And this is just a variety of the ways that maize is stored. And it spans everywhere from on the cob, literally on the ground next to the field under a black piece of plastic. I don't know if you can see it in that picture, to stored very neatly in the home, to stored kind of haphazardly in an outbuilding with their chickens running around on top. They store it off the grain in some areas, and actually all these little black dots that you see are the maize weevil coming out of this grain. And this grain is still for the family. It's not for their animals. It hasn't been damaged enough that it's been set aside for the animals. So this is what people are eating. And then there are farmers that have access to silos. And in this picture in particular was a co-op, and they actually had multiple silos and hermetically sealed bags because they were selling to restaurants. But some of the individual farmers that had silos, they were older, they weren't hermetically sealed anymore. And so you could still find maize weevil even in the silo. And this is what the damage looks like. On the left is this picture of a cob that's really been decimated by the maize weevil. You can see all the holes where they came out. And that is really not fit for human consumption anymore. And that would probably be fed to their animals. And on the right is what's really left at the end of all the damage. And all these black dots are weevils. And then this flower looking stuff is a mixture of the frass, or poop, from the weevil and corn flour. So it's really not usable at that point. And so this damage can be pretty severe. So I first got interested in maize weevil during my PhD work when I was part of an IGERT fellowship, which is an interdisciplinary training grant. And we went to Mexico because we were charged with considering the use of genetically engineering insect pests, or agricultural pests rather. And Mexico is a really interesting place to think about this because although Mexico does not allow genetic engineering of maize because it's the center of origin, because it's so culturally important, and they're trying to protect their land races for a variety of reasons. Genetically engineered maize is not allowed, but genetically engineered other crops are. So soy and cotton are allowed to be genetically engineered. So there's this really nuanced view of genetic engineering and its use in Mexico. So we were thinking along the lines of, well, maize weevil is the most common pest that we're coming across when we visit farms. Would farmers be interested in using a genetically engineered pest so that they don't have to use a genetically engineered maize to protect their food for the year? And so we were originally thinking about that, but during the course of my research, I realized that we are so far away from being able to genetically engineer a maize weevil because there were so few genetic resources available. There were literally 9 microsatellites, a couple of mitochondrial markers. There was a very helpful karyotype. And beyond that, there were primers for specific genes, but none of those genes were really genes of interest for me. And so I set out to start to develop genetic resources for this species. The first thing I needed to do is figure out the genome size, because if you're going to do any next generation sequencing, you have to know the genome size so you know how many individuals you can put on a lane to sequence so that you can get the right amount of coverage. And so once I did that, then I wanted to create a linkage map so that we could have markers, genome-wide markers that could be useful for other projects. And then because I am interested in evolution and population structure, I wanted to see how much structure existed in populations of maize weevil in Mexico, because that's also one of the things you need to know before deploying a genetically engineered species is who it's breeding with, how far is its breeding range, and so that you can predict how it genetically engineered insect might move in the environment. So I started with determining the genome size, which is actually a fairly easy assay. All you do is stain neural cells of your species of interest with neural cells of a known species, in this case, Drosophila, and you compare the signal intensity from a flow cytometry, and then you can use this simple formula to figure out the genome size. And so we knew that the species has an XY sex chromosome. And so females had a slightly larger genome size of about 681 megabases, and males was slightly smaller because of that Y chromosome being smaller than the X chromosome, and it was about 674 megabases. So once I knew that, then I could design a project for next-gen sequencing. And this was to develop the linkage map. So I obtained 2 lab colonies from the USDA, one from a lab in Florida and one from a lab in Kansas. Now the Florida labs colony originated from Tifton, Georgia, which is a little town just north of the Florida border, if you're familiar with US geography. And that colony was started in about 1961. Now, we didn't have any origination information for the colony from Kansas, but we knew it had been in the lab for a number of years and had likely diverged from this other colony. So I used these 2 colonies to create a breeding cross where I mated a female from Florida with a male from Kansas. Then I mated their offspring together to create an F2 population. And I put the parents and the offspring onto a lane of Illumina HiSeq using the DD-RAD method. If you're interested, I used MSP1 and EcoR1 restriction enzymes. And I was aiming for about 20x coverage of the offspring and about double that for the parents, just so that we would be assured that we had enough coverage of the parents to create a catalog of SNPs. And you can read through the kind of simple stats that we got from our run, but 4 of the individuals of the offspring did not sequence very well, so only 104 individuals of offspring went into the map. And then I used the STAX pipeline with pretty stringent Mendelian filtering and got about 1,100 SNPs that I then used into the program OneMap with a Konsombi mapping function, and all but 2 of the markers map to a linkage group. And using a LOD score of 11, I was able to get back 11 linkage groups, which corresponded really nicely to the karyotype that has 10 autosomes and an X chromosome. We weren't able to get back SNPs on the Y because there was only one Y in our mapping cross. And so I was really pretty excited with the way this turned out. We have lots of markers spanning a good, the whole genome. The linkage groups looked really nice. And so I was pretty pleased with this as being a resource for the future for maybe sequencing the whole genome or using these markers for some other study that might be of interest to someone. And so during this time, I of course, have always had this interest in population structure. And during this time, a paper came out from a group in Brazil. And now there are really like 3 labs. There aren't that many people that work on maize weevil. So this group in Brazil is like one of like the best labs looking at maize weevil. And they use those 9 microsatellite markers. And they found that there is high gene flow throughout the world in maize weevil. and that this indicates a recent range expansion, which makes sense from what we know of how corn and when corn left the New World and went to the Old World, and when this relationship between the two species likely evolved. But the biology of the maize weevil doesn't lend itself to thinking that there is high gene flow throughout the whole world. So if you're thinking about subsistence farmers in Mexico and subsistence farmers in Asia, they're likely not trading grain very frequently. And we know that maize weevil also, they're not strong flyers. They can fly about a quarter of a mile, half a mile. So they're really being transported through the grain trade. So I hypothesized that, well, 9 microsatellites aren't very powerful. And so they're good at picking up this biological event that happened a few hundred years ago, but they're not powerful enough to pick up strong, more recent differentiation. And so I went back to Mexico and sampled 12 different villages in both Oaxaca and Chiapas. And these were chosen partly because of the wide geographic ranges in these states. They're both mountainous regions and coastal regions, so big difference in altitude, but also there's this isthmus where there's a lot of wind energy and wind farms. So if you drive through here, you just see acres and acres of windmills. And so this really windy area is a physical barrier to migration for the species because, again, they're not strong flyers. So if there's gene flow, it's through the grain trade here. And so these are where we sampled. And I actually went with another grad student, Mike Jones, pictured here, with me, and he was an economics grad student. So he was really interested in the farmer's decisions for when to store and when to sell grain, and more of these social factors that go into how the farmers are treating their maize. And I, of course, was interested in the genetics of the maize. And so I would go and collect the weevils, and he would interview the farmers. And we made a pretty good team and were able to cover a lot of ground in just a few weeks. And so when we came back to the US, I had this great undergrad who's now a grad student here at NC State, Destiny. And I charged her with genotyping a bunch of these weevils using those same microsatellites that the Brazil group used. And we found, as we expected, there not to be any structure. Everything is piled up on top of each other. But this is really just to check, to prove to ourselves that, yeah, I mean, this makes sense, right? But because we had this dd-RADseq protocol, I was able to sequence 2 lanes of samples. On the first lane, I put the 4 populations, 2 from the highest elevation villages and 2 from the lowest elevation villages on one lane, and from each state on one lane, and then samples from the remaining 8 populations on the other lane. And what I found when, after I did the bioinformatics work, was that we are getting these 3 strong groups, but when we looked at it more closely, we have extreme lane effects. So lane 1 is all grouping together, lane 2 is all grouping together, and then this third group actually turns out to be samples with missing genotypes. Now, these are not samples that did not sequence. All of these sequence just as well as the samples in the other populations, but for whatever reason, the parameters that were chosen during the bioinformatics pipeline, the SNPs that were retained there, were not found in these populations. And so that, probably could be a biological reason for that. Maybe these are more differentiated than the others, and so they're not sharing as many common SNPs, or maybe there's some other technical issue there. So I just removed those populations with the missing genotypes and looked at the remaining 8. And when I plotted the PCA after doing that, these are the same data. you can see that we can still see the lane effects a little bit, because these three populations that are stacked on top of each other are from lane 1, and the remaining populations are from lane 2. And just to orient you, these are color coded by the location, so that the colors on the map match the populations that you're seeing on the PCA. And so I said, okay, well, PC1 is explaining these lane effects. So let's plot PC2 and PC3 and see what is going on. And here is where you can finally see population differentiation. And it's, you know, I was pretty pleased with this. And what we're seeing, we have these 4 populations that are really distinct from the others. And then there's this group of 4 populations that are clustering very tightly together. And just anecdotally, these 4 villages are the most isolated, both socially and geographically. And these 4 villages are all located in Oaxaca and had easy access to the main highway. So they were much more interconnected socially and physically than the other populations. And in addition to that, PC3 is actually picking up on this geographic difference between the 2 states. So this orange population and this green population are both from Chiapas, where these other populations are from Oaxaca. And so I was really pleased with this. And although our FST values are still low, so there is still a lot of gene flow, we do see that some population structure exists, and we were able to pick up on that using these SNPs, which of course, thousands of SNPs are going to be more powerful and able to find these differences than using just a small number of microsatellites. And so this work I did for my PhD, I figured out the genome size, which was, you know, important for next-gen sequencing. I produced the first linkage map, high-density linkage map for the species, and I was able to identify population structure using dd-radseq. And through that, I was able to show that you can actually control for lane effects with very careful filtering and analyses. And so although I'm not currently working on Maize Weevil, I would like to see the field progress and maybe, you know, create a genome for the species. And, you know, we could look for areas of the genome that are under selection using those SNPs that were in the linkage map. And I would like to further explore population structure at a larger geographical scale. So I actually have samples of weevils from South Africa and Brazil and Mexico, obviously, and a couple of other locations that, you know, if I could get the funding to do another study, I'd love to take a look at that using the dd-radseq. I think we'll see some differences from that previous study that was published. And so with that, I need to thank all the people who helped me collect samples and get to Mexico and process samples in the lab. And then, of course, funding sources, which make all of this work possible. And here are pictures of everyone who helped in the lab over the years. And I can take any questions. Thanks for your attention.
Okay, thank you very much, Jennifer. I had a first question. Is like, so you began your talk by saying that your goal was to engineer, right, the Weevil.
Well, the goal is not.
I was wondering, like, what did you have in mind? Like, which type of engineering would you like to make, or, you know, what was your?
Yeah, so I guess my personal goal has never been to engineer, is more to see if farmers would be willing to consider that as a method that they would be willing to use. But, you know, in the case of engineering, I think the most obvious and probably the least risky approach would be to use a population suppression technology, where you release, say, like they have a mosquito, the riddle mosquitoes that are pass lethal, sterile lethal genes on. And so the insects you release will mate with the wild insects, but they will not be able to produce offspring. And so your population size will actually decrease over time. And that would, of course, save the grain, because the weevils would not be, there'd be fewer weevils. But yeah, I don't think anyone is seriously considering doing any of that in the near future.
Okay, so there is a question by Jeff.
Yeah, hi, that was fun, thank you. I guess I'm just wondering your speculation. I logged on a little bit late, so apologies if I missed it. How the weevil first encountered maize? Was it when maize moved to China, or did the weevil move to Mexico or some other part of the world?
Oh yeah, I think it's probably when maize moved to the weevil. So let me see if I can find this slide again. It's up in the beginning. I have to move the chat here. So this map, which I took from this paper, shows when maize left and went to Asia. So the 1500s to the 1800s. So I think it probably happened in Asia and then grain trade, but no one has done that work, so that is total speculation.
Cool, thanks. Yeah, I was thinking that looking at the diversity of genotypes on maize, you might be able to answer the question.
Yeah, that was one thing I was interested in. So I did during that study take down the land races or that the farmers were growing, but a lot of people just called it like Blanco, but I don't think it was always necessarily the same Blanco. So if I could go back, I would probably also try to take samples of the maize to get, so we could get a genotype profiles from what was actually being grown in each village.
Okay. Sanzen, do you have something?
Yes,
Yeah.
a nice talk. And I just saw your PCA plot that you identify a group of individuals with low number of genotype data. And you said that the number of reads actually is high. And that could be, if using reference genome for the pipeline, it could be caused by the genome are very different between this group and reference genome.
Yeah, it could. So I actually called the SNPs de novo because we don't have a genome, and I wasn't at that time using because I was kind of doing the work in parallel with the linkage maps. So I didn't actually have the linkage map ready when I was doing that. So all of those SNPs were called de novo from the sampling, but so they were not mapped to a reference genome. They were mapped to the parents.
You get a lot of reads, and where those polymorphism or where those reads gone? I mean, why no marker associate those individual if you have a lot of reads?
So I just want to make sure I heard you correctly. So why did they not have markers associated with those individuals? If what? I couldn't catch that last part.
You have a lot of reads for each individual, right? And why they could end up no mark for those individuals?
Yeah. So in the bioinformatics pipeline, it could be that those populations are so different from the other populations that they just don't share a lot of the same SNPs. And because the SNPs were chosen from the population de novo, and we were looking for, specific, there are lots of different parameters that you're picking in the pipeline. I could have gone back and redone it using different parameters. And if I was doing that now, I probably would have done that to drill down into exactly why. But at the time, I was so focused on those lane effects that I was like, oh, maybe let's take those out and the lane effects will go away. But
could be very
yeah,
interesting for that group.
it could. I'd love to have the time, or maybe a student, to dig into that data some more.
Yeah, good. Thank you.
Mhm.
Matt?
Yeah, I was just curious if you'd heard folks in Mexico or seen any evidence that the maize weevils also using the wild relatives of CI, or the wild relatives also have this fruitcase that surrounds the kernel, so maybe that acts as a barrier.
Yeah, that's a really interesting question, and not something that I looked at all. What I do know from the literature is that and honestly, my knowledge of the wild relatives of maize is very limited. But in the literature, when people are looking at, say, the qualities of different land races, because some land races are more resistant to maize weevil infestation than others, there are certain chemicals that are present, like phenols, that are present in certain land races at higher quantities that act as a natural pesticide. Also, the density and how hard the kernel is can be a physical barrier for the female to, chew that initial hole to lay the egg. So I'm not sure if the wild, like with the hardness and the chemical composition of the wild mazes to, what's being grown for food now. But those are some things that are present in land races that have been associated with greater resistance to infestation.
Cool, thanks.
Okay, is there any other question for Jen? If not, I think we can end up here, and we'll see you all not next week, right?